All right, let's turn to our next session where we're going to be taking a look at recent clinical trial outcomes in Huntington's disease. We're going to take a look at some of the lessons that have been learned and how that's shaping future research directions, and also why there is a growing sense of hope within the HD community. Would you please welcome to the stage Professor Clement Loy.
Thank you, Amelia. And I would also like to acknowledge the traditional owners of the land on which we're meeting today. I tried to say it right. The Ghana people, is that right? Yeah, and pay respect to elders past, present, and emerging. Thank you very much to Lenny, to Chris, and also to Ali for the invitation to be here today for two reasons. So first, it's nice to see so many old friends and new friends. I don't think we've had a meeting like this for over 10 years now, so well done for organizing it. The second thing is a topic. So I've got a little secret to tell you. For the past 18 years, there hasn't been a date, I don't think, about Huntington disease trials. So this is about 10 years after the first gene-lowering dosing, so it's a really lovely opportunity to think through it. So I hope you find it interesting. Acknowledgements. First, a huge thank you for all the people that had contributed to the HD trials. Put your hands up if yourself or a family or friend have been in Huntington disease study of any type. People that don't have your hands up, could you clap? Those studies couldn't have happened without the generosity of the families, and this talk is for them. A few disclosures. I've been an investigator for Enroll HD, Roche, Wave, PTC, Novartis, and Sarepta, and my colleague, Associate Professor Florence Chang, is a investigator for Skyhawk. So at Westmead, we had been part of almost all of the trials. I have not received any personal remuneration for any of those. I'm also a chief investigator for two other studies I'll tell you about, funded by the government, the Pre-HD NAC trial and also the We Hear Your Voice study. Last, I've been involved in regulatory work. I've been a member of PBAC and TGA now. What I'm talking about now is personal and professional opinion of my own and not related to my roles there. So in the next 25 minutes or so, I would like to first talk about Huntington lowering strategy. You've had two eloquent and really up-to-date examples of those. I would like to bring them all together as one concept and think about, what do we need to see for these strategies before we can bring it to the clinic. Hopefully they'll give you a framework that help you think about other trials as well. Then after that, I'll look through all the results that have been publicly accessible to date and think about this strategy as a group. Then I'll touch on a few studies that are happening in Australia at the moment. There's actually a lot happening. Later today you see Mel talking and Sally talking, but I'll just touch on a few and then a few concluding remarks. Okay, so what's the Huntington lowering strategy? Who did biology at school or uni? Yeah, do you remember? I won't quiz you for it, but we always get taught, you've got, it's called the central dogma. You've got DNA that get transcribed into RNA and then translated into proteins. And this assumption that, if there's a problem with your DNA, then it makes a bad protein that gives you disease. I think for Huntington disease might be a little bit more complicated than that, but that's not a bad way to start thinking. And the Huntington lowering strategy as a group aim to change the course of disease of Huntington disease by lowering the Huntington protein rather than treating the symptoms alone. And a number of these are RNA-based technologies, and now I'll group them for you to see how they fit together. So they're antisense oligonucleotides. The first one is from Roche tominersen. In detail, this is how it works. You can see in the diagram there. Sorry, I can't point both sides at the same time, but you've got a gene, you've got transcript into RNA, and you make protein. Now, we all have 2 copies of each gene, one from dad, and with Huntington disease, having a mistake or elongation of the CAG repeat is enough to cause disease, and one of these would make an abnormal protein. What the oligonucleotides do is they're designed to attach to the CAG expansion. That means that it attaches both the abnormal as well as the normal copy of your transcript. And then they are attracting an enzyme that eats it up, and as a result, you lower both the abnormal and normal protein. Roche and Wave are both, the first generation Roche are both antisense oligonucleotides, as well as the new one that you've just heard about. And Wave and the new generation one are specific to one allele or the expanded protein copy of your gene. So the other Huntington lowering strategies are small interfering RNA, which again works a similar way. It attaches to expansion and then activates a different mechanism, then lowers the protein. A group that's quite different are the splice modulators, and that's for PTC, which is now Novartis trial, as well as Skyhawk. These are modulators that go and actually act on all the transcripts in your cell and change how it's processed, called splicing. And as they're spliced, oops, I'm going to go back. As they're spliced differently here, then sometimes a stop signal is spliced into the transcript and then stop making the protein. And one interesting characteristic of that group of molecules are that they can target more than one target at a time. And you have heard about the PMS1 story then. And another one that a lot of you might have heard about in the news is the Unicure system where you use an inactivated virus to deliver microRNA. Again, that interferes with the RNA processing system. So in fact, all these drugs, all the things that you hear about are all Huntington lowering strategies. Does that make sense? Yeah, okay. So now, if you can do that in the laboratory, what do you want to see before you take it to the clinic? And these are the things. First, you need to want to know that it's feasible to run a trial for people with Huntington disease because it's a relatively rare disease, and you've got to have some clinical readout that that would give you the result in a reasonable amount of time. You want to know the drug that achieves the intended biological effect. You want to know that the drug is safe. And then you want to know that the biological effect that you can see also correlates to clinical gains, and preferably against a placebo in an RCT type randomized controlled trial setting. After that, you might consider any variation to the approach, and you had 2 good examples of it, being allele-specific with the new Roche drug and being Skyhawk with the second target. And finally, you think about what's the right dose and what are the times for drugs. So has anyone been following all the drug trials? Yeah? Yeah? What's your feeling about it? Have we got anything yet? Didn't think so? No. Yeah, so actually, I mean, we've had news about these early trials kind of haven't got a drug to come to clinic yet, but I think those trials have taught us a lot about what we want to know. And for the next section, I'm going to show you some of those trial results and how these trials have actually answered some of these questions for us already. Okay. what have we learned? I think many of us might remember when the first Roche trial came, there was a huge amount of excitement, and it was a little bit sad towards the end that it had to be stopped. I'll just briefly go through this. This is a scale, a clinical scale there, going vertically and time. This was the first Huntington lowering drug that was trialed starting in 2015, I think. And unfortunately, the high dose people did worse than the placebo people. Now, despite that, there's actually a lot that we have learned from it. The first thing is that trial was designed based on observational studies such as Enroll, that's running now, and Cohort before, and Registry. And those natural history studies have taught us a lot. The second thing is, even though we saw that there's some differences between the groups and not in the direction that we wanted, we also noticed that the clinical measures, the outcome measure we have in the trial are precise enough that you can actually detect a signal over a short time. So in other words, we've got the tools to run a good trial. And it recruited 791 people in 15 months in a really rare disease with a strict inclusion criteria. So I think this is really a tribute to the generosity and resilience of HD families and how the HD community cooperate across the world in clinical studies. So, question, is it feasible to run an HD trial for Huntington lowering? I think definitely. So we've got green light from that. So the first question we've got answered. The second question is, does this approach achieve the biological effect? What was the biological effect we were aiming for? Is the Huntington lowering strategy? Well, we do want clinical improvement, and we thought we might be able to achieve that. Hello, we've been sitting for years, Alice. It's good seeing old friends and new friends, and the approach here is we think the abnormal protein would also be lowered, and actually that's been achieved in just about every trial that we have seen so far. It's happened in Roche. It went down 20 to 40%. It's happened in PTC. It went down 20 to 30%, and in Unicure as well, which thought to have gone down 50 to 60%. So the question is, are these drugs doing biologically what they were designed to do? And I think the answer is also yes. So we've got two questions answered now. Then the question is, are these drugs safe? So, safety of drugs depends very much on the individual candidate, but I guess the question is, are there instances where this is safe? If it's unsafe in every situation, then maybe lowering Huntington is a bad idea. And there are at least two examples. The PTC or Novartis drug has got very comparable adverse effect profile across the different arms, including placebo. And similarly, AMT, which is interesting, Unicure, I think it's interesting to note what happens there because even though it's a single arm study, that's been going for 6 or 7 years and they haven't seen anything bad either. So I think, it's probable that, Huntington lowering strategy is safe or can be safe. So what's the next question? Well, is the biological effect lowering the Huntington protein actually, does that translate to clinical effect like what Liz said? I think that's a critical question. And did anyone hear on the news about the Unicure story? No. Yeah, that came up on the BBC amongst others. It's a study that was in the US and UK. And basically, The study started off as a randomized control trial with placebo group, then became a single-arm study, and then the single-arm study has got nothing to compare against, and that's why it was very hard to draw conclusions. And I think it's important when you look at whether there's any clinical improvement that you've got a placebo group, that's randomly assigned and is blinded. Now let me do a little experiment here to demonstrate randomization. Put up your hands if you grew up or have a dog at home right now. Do you notice on this side and that side is about the same number of people? I'll try another one. Put your hand up if your month of birth is an even month. Look, that's pretty even as well. So that's the power of randomization. People sat down randomly, not knowing whether they've got dogs or not, what month. And randomization allows you to adjust for all the unknown things that are not measured. And the problem of not having the randomized group for the Unicure trial means that no matter how you adjust against it, there might be things that you can't take into account for. So, having said so, there had been one study, at least, that showed a trend of lowering protein means better clinical outcome, and that's from the PTC trial. It's small numbers and a subgroup, but at least it's going the right direction. In some of the earlier trials, protein was lowered, but people didn't improve. And they also have some longer-term data. This is interesting because even though it's an extension study, people remain blinded. So the group with the higher dose seemed to suggestion that it could be doing better than the lower dose. I wouldn't use the historical control, but that is also a bit of a signal. So do I think that lowering the abnormal protein could have clinical consequences? And I would say maybe. We're not there yet, but we may be. That there is a signal that could be there. So in my list of questions, the last two are one, thinking about variation to the main mechanism of action. And actually, you've had two wonderful talks for that already. You can be allele-specific, only looking at the expanded allele copy of the gene or RNA, that's the WAVE and the new Roche study, or you can target a second target, that's PMS1. In terms of right dosing and timing for the drugs, that's to be determined. So back to our list. Well, we haven't had the drug in the clinic yet, and we don't have a really positive trial, but how many of these questions have we answered? One, is it feasible? Yes, green light. Is there a drug that do these drugs achieve the biological effect? Yes, green light. Can these drugs be safe? Probably, there's at least some circumstances. Is that biological effect accompanied by clinical improvement? There seems to be a hint of it. There's at least one potential example now, and then we're thinking about variation of the mechanism. So I think we've come a long way, despite what being disappointed that we don't have a drug in the clinic. Okay, in the next few minutes, I'll just quickly go through a few other studies. In Australia, as I said, there are a lot of other things going on. Answering the question that came in earlier, there will probably be a trial for people with more advanced disease on predopidine. But I'll just show you a few examples of studies that I've been involved with. One is Enroll-HD. I think many of you know this. There are now 20,000 people in the study. It's gives us really important information to design trials, and that happens at Monash, at Perth, and in Sydney at Westmead. The federal government has funded us to run this pre-HD and its acetylcysteine trial. This remains the only trial in the world that's purely for people without symptoms. Where it came from is that they were making mice that have changes in their amino acid processing pathway. And notice they look a bit like Huntington disease mice. And Tony Hannon, who's in Melbourne, then started feeding the HD mice with supplements and realized that it could actually slow down the shrinkage of part of the brain and delay the onset of movement problems in those mice. So for that reason, we have designed a randomized control trial for people tested positive but no symptoms yet. It's a blinded randomized control trial, 1 gram of N-acetylcysteine twice a day versus placebo, movement and MRI scan as primary outcome. It's a 3-year study. It's a pretty light study. So after the 3 months, first 3 months, then it's just 6 monthly visit. If anyone's interested, we are very proud to have a number of sites now. Alan and Karen runs one in Adelaide. John O'Sullivan and John Adam runs one in Brisbane. Melbourne, we are still discussing. Perth, Carolina, and Mel's there. Randomised the first person today. Yay. And Sydney, Florence, Sam, and myself run the site. For people that have got more advanced disease, one thing that came to mind is, does anyone use Siri or Alexa? Yeah. What would happen if you give Siri to someone with Huntington disease? You probably couldn't quite understand it. So we're developing alternative algorithms so that Huntington disease speech, if you like, could be recognized, and that would open a pathway to other assisted technologies. So these are for people with more advanced diseases. Okay, a few final comments. So we don't have a Huntington lowering therapy in the clinic yet, but over the past 10 years, we've established that these trials are feasible. We know that we have got examples of drugs that achieve the biological effect. They can be safe, the examples of it, and the early signs suggesting clinical effect. I think we've come a long way. In parallel, there are many active trials and studies in Australia, and also NDIS has also made a huge impact. So I think there's much hope. I just want to leave you with one picture. Do you think it's half full or half empty? Hands up. Half empty? Oh, half full? Half empty? Now that wasn't randomized for some reason. But I just want to show you, this is how I think about it. It is half full, but there's a lot more water being poured into it right now, so it'll be full before long. So thank you for listening, and looking forward to chatting further.
That was an incredibly engaging conversation. Before I let you walk off the stage, Professor, are there any questions? We, of course, will be hearing from the Professor a little later on in the program too. Just one right here as we scurry to get a microphone over. I might hand this over to you if
Yeah, of course.
with it.
Thank you. Thanks, Amelia.
Thank you, Professor Loy.
Pleasure. Oh, Clement.
Just a
Clement.
short question. You talked a lot about placebo, and I wondered what your attitude was regarding natural history versus placebo, and maybe what the TGA's attitude is.
Oh, I can't speak in the capacity of the TGA, I'm afraid. But, I mean, I can speak as a clinician in this field. If you go back to 2008, 2009, 2010, some of the early simple molecule trials, the problem we had was placebo, because placebo effect's pretty strong. So if you've got a study that doesn't have a placebo and you use natural history data, there's likely to be a difference, because the scales that we use do depend on people's cognition effort in motor function, and also, the clinicians could be biased in measuring. So I think placebo and randomized control trials are really critical. I know that there is some move towards maybe we can have uncontrolled data, but I think I'm still personally yet to see a good example of that. I don't know whether that answered your question, and I would love to chat further after this.
I've probably got a loud enough voice for those who know me. For the online people, I'm probably not loud enough. Clement, thank you so much on behalf of Huntington's Australia. It's a pleasure to always chat to you. I'm just really interested, every time I meet Clement, we have amazing, and we get the chance to catch up, amazing conversations. But in the theme of today, and not wanting to point out your longevity in this space...
You're saying I'm old.
No. But maybe just in a few sentences, what does hope look like for you now, in your experience in this space and where we are today in 2026?
I think hope looks like how far we have come since, I first worked with Liz McCusker and had the privilege looking after families with HD in 2003. We have come a long way. I mean, NDIS has made a big difference. We are more mindful what services can do. For example, inpatient rehabilitation can improve function. I mean, when I started, that was never really thought of. And when I started, there might be one trial at a time. Now we've got 6 or 7 going at a time. We do our bit here, and then we go to sleep. Germany does their bit there when they go to sleep. I think the future is incredible. There's hope.
Thanks very much, Clement. With the Roche study that the lady whose name I can't remember, of course, they are just targeting the abnormal, for want of a better word, gene, so that the normal gene can produce HTT. But with the Skyhawk one, it's both genes, isn't it? So if you got to the stage where you completely suppressed HTT, would that be bad? Do we need it? I guess we need normal HTT in our brain. Do you have much to say about that?
Yeah, that's a very complex question. So, for example, in animal models where you've got 2 copies of expanded CAG repeat, the animals don't survive. But in people, for example, I've looked at a number of people who've got 2 expanded copies, and they don't seem worse than the others. I think the jury is still out for that. And there's also an element of how much you suppress both. So obviously, if you suppress 40%, it's different. If you suppress 80% of the normal. Normal Huntington does have some function, so that's still unanswered. And there has only been one select allele specific trial before the Roche one. That's the wave one, but for that one, it didn't manage to shift the protein. So it could be a problem with dosing as well. Hi, someone put up a hand? Okay, fantastic.
We'll leave it there. Thank you so much, Professor Loy. Clement.